Hello, everyone. Welcome to this webinar, a webinar dedicated to the improvements of sample preparation with the 3D bead beating technology by Bertin Technology. So thank you for joining. Well, I'm sorry, we are just a few minutes late. I had a few technical issues. I hope it's going to work perfectly for the rest of the session. However, let's get started. So just before starting, I'm going to introduce myself. My name is François D'Hubert, and I am the Precellys French product manager. So I will be your speaker today. So do not hesitate to ask your questions in the question box that you have on the right side of your screen, probably. During all the session, your microphones will be muted. So write down your questions, and we'll have a question and answer session at the end of the presentation. So to go back to the presentation, so a small agenda of what we are going to see today. So briefly, I'm going to make you a small introduction of Bertin as a company. And then we'll move on the main topic of this session, which is the challenges of sample preparation and the way that you can improve your sample preparation. And finally, the question and answer session that I've been talking to you just before. So as an introduction, we are going to see Bertin Systems and Instrumentation, so the company that I'm working for. So we are involved in different fields, as you can see, very various fields, defense and security, where we cover, well, across different technologies and mainly dedicated to optronics and sensors of different kinds in order to monitor the sites and to save sites and people. We also have a business line dedicated to nuclear and physics health, where in that case we provide some probes dedicated to radiation measurement, again in order to protect people and sites. We have a department dedicated to space and big science, where we have high performance optical and optomechanical systems. We have a very recent example of what we can do since, as I'm sure you all heard of it, a small robot landed on Mars a few days ago, and the camera that made the images has been developed in part by Bertin Systems. So we are pretty proud of being on Mars as well. And if we go back to Earth, we also have another department called Hospital Waste Management, which is dedicated to the banalization of medical waste by a treatment on site of the medical waste in order to prevent the spread of contaminated waste in the environment between the production site, usually the hospital, and the treatment site. So, so this is quite interesting in terms of environment in that case. And finally, the life science business line in which we and I work, which is dedicated to laboratory equipment, kit and reagents. And so if we make a little focus on that range, we have four different product range that cover more or less a global workflow that we meet in research. So starting from the collection, thanks to a range of instruments called the Coriolis. So we have two different kind of instruments that basically collect the air and are able to trap the components that are present in the air. So biological components mainly. So this is quite trendy as well at the moment. The COVID-19 virus spreads in the air, and so we have a lot of studies that speak about that studied thanks to the Coriolis, the spread of the virus in the air. If we move on in the workflow, we have the sample preparation, which is the topic we are going to cover. So I will develop later on, thanks to the Precellys range. And then we have the other side of the workflow, which is the analysis, thanks to bioreagents, mainly based on immunoassays and ELISA kits, and also a range of products dedicated to observation, thanks to our knowledge and our know-how, in optronics. We developed a microscope, a digital microscope, in order to facilitate the life of cell culture facilities. So it's a digital microscope, an inverted digital microscope, on which you have onboard applications, just like on your smartphone. So for cell confluency, cell measurements, things, kind of parameters that you mainly monitor during the cell culture process. So if we go back to the main topic today, which is the sample preparation and the challenges associated to sample preparation. So if we still remain in the workflow approach, well, in all biological experiments start with a biological sample. This biological sample is usually quite complex. It can be a full organism, or it can be a part of an organism, or just cells. But in any case, it's not pure molecules. And these biological samples are meant to be analyzed for different purposes. So they need to be prepared in order to get transformed from their native form into the final analyzable form. If we just give a look to the final analysis, it can be very different, starting from the same material. So sample preparation needs to cover a lot of challenges depending on what the final analysis is, but all these final analyses have a common point, which is they require more and more high quality molecules. Indeed, in the past, well, the analysis was a little bit more macroscopic, or was just related to the presence or absence of some molecules. But now what we see is that more and more the analysis is dedicated to a deep analysis of the molecules themselves. So if we consider, for example, qPCR, we have the quantitative aspect of some genes, or if we consider mass spec, we have a deep analysis of the structure of the molecules, the identifications of molecules. So we have a lot of techniques that require high quality molecules. So the real challenge is to be able to reach these molecules and to keep them in a good way, in a good state, in order to be analyzed. So we usually divide the sample preparation in two different steps, which is the homogenization and the purification. Well, in people's minds, purification is usually considered as being the most important part, as this is where you are going to collect and purify the molecules you want to work with. But we at Bertin Technology consider that homogenization is also a very, very important step, because it ensures a good way to get access to those molecules, and there are also different aspects that need to be considered in order to get high quality molecules. So if we go to the sample preparation challenges, Obviously, the first challenge will be to break down the sample. So starting from your biological material, you will have to be able to break it down, to homogenize it in order to extract, to free the molecules of interest that you want to target and purify afterwards. So this is obviously the first challenge. Then the hands-on time need to be considered during sample preparation. If you have a lot of samples, you don't want to spend a lot of time, mainly because if you spend time in treating the samples, there is chance that the first sample that you are going to treat will not have the same behavior as the last one. So in order to have reproducibility, which is another point that we'll see later on, you need to have a good hands-on time for the sample preparation. Cross-contamination, of course, is something that everyone wants to avoid because, well, you need to make sure that what you are going to analyze in your sample is the sample itself and not one sample that you have been treating previously. Reproducibility, as I was saying, a very important aspect. You want to make sure that from an experiment to the other one, you will have the same level of reliability in your results. So if your method is reproducible, you are sure that what you made will be comparable from one experiment to the other. And finally, the yield and the quality of the molecules you are targeting. So of course, getting enough molecules to make the final analysis is a crucial point. And getting these molecules in a good state is also a crucial point. So you can have plenty of one molecule. If it's degraded, you're not going to make anything with it. So these are the two other points that we'll see in the end of the presentation. So if we just focus on the first challenge, which is breaking down the sample, it seems obvious, but there are different ways to treat samples. The most common one is the mortar and pestle. So this is a technique that has been used for thousands of years, not only for biology, but also for other purposes. But in biology, this is a classical method to homogenize samples. The main weak point of that technique, despite the fact that you can treat samples from the softest ones to the hardest ones, depending on the strength of the users, is that you have a variable efficiency and a quiet law, which again depends on the user. So being a manual method, if the people who handle the mortar and pestle are very strong, you will probably not get the same efficiency as someone who doesn't have any strength in the arms. Another way to treat the samples is to use a blender. So here we have an instrument. So in terms of efficiency, it's a little bit better, but there are some limitations in this technique. As soon as if you are using the blender to treat very hard samples, you may risk to damage your instrument, your blender, the blades that homogenize the sample, and so you will not be able to treat all the samples. And the final technique, which is also very commonly used, which is sonication. So it's another instrument. The principle is to put a probe that will emit some ultra waves, some ultrasounds in the sample, and by vibration you are going to break the cell walls and the cell membranes. So it's very efficient on cell culture, but when you are treating more complex samples like organs already, you are going to reach some limitations. So the efficiency decreases with the complexity and the hardness of the sample. And that's why there is bead beating. So what is bead beating? Basically, you put your sample in a tube, so a classical lab tube, microtube, and associated to this sample, you are going to place some beads, some material we call lysing matrix, and those components being in the tube, you are going to shake the tube, and so the beads will hit the sample and homogenize it. It's quite simple, and there are different ways to do it. What we call the do-it-yourself version, which is a manual process. So in that case, you also have to be very strong, but more often it's made with a vortex, with a bench vortex. In that case, the efficiency is not that good, but it can work. Then you have what we call 1D bead beating instruments. So in that case, the tube will be shaken from one side to the other, so commonly in the length of the tube, and so by agitating quite vigorously, the instrument is going to homogenize the sample by the mechanical action of the beads on the sample. This has a small limitation because by moving the tube always in the same direction, the sample tend to accumulate in the bottom or in the top of the tube, meaning that it's nearly always the same surface which is exposed to the beads. However, it's quite efficient. And then we have what we call 3D bead beating. So in that case, the tube is not shaken in only one direction, but has a movement which is in 3D. And so the beads and the samples inside will move in a random way. In terms of efficiency, there is a real difference between 1D and 3D bead beating, since as I said, the beads will move randomly, and so they will hit the sample randomly. And in that case, it's not always the same surface of the sample which is exposed to the mechanical action of the beads. So in that case, you have, by having a random movement, you have a random homogenization on your sample, and so it's much more efficient, and it takes less time, which is quite important in terms of sample degradation, but we'll discuss that a little bit later on during the presentation. So if we sum up and compare the different bead beating techniques that there are with the most versatile technique, 1D bead beating and manual bead beating allow to treat quite hard samples with a quite good efficiency, but again, the most efficient is the 3D bead beating technology, which ensures an efficient homogenization of the sample for every kind of samples, whatever the hardness is, and whatever the size of the sample is. So very good efficiency with this technique. If we consider the other points that I listed in the introduction, so we are not going to discuss all these points, but just by quickly look at the table, you can see that in every case, the 3D bead beating is the most efficient method. So in terms of reproducibility, it's much more interesting than a manual method, of course, but also if you compare it with other instruments. Cross-contamination, of course, there is no risk as the consumables are single-use. And of course, the hands-on time is also much more interesting, as you can treat a lot of samples at the time, so up to 24, which is not the case with the other techniques where you can only treat one sample at the time. So time-consuming. So this is for all these reasons that 3D bead beating has become a gold standard for sample homogenization, because it brings all the benefits that we listed previously, adaptable to any biological sample, no risk of cross-contamination, time saving thanks to the multiple samples treated at the same time, and very efficient, which brings a better yield recovery for the molecules of interest of your sample. And of course, the reproducibility, as it's an instrument. When you make a protocol on your instrument, you are sure that it's going to make the same protocol every time you use it. So here is the range of instruments that we have developed. So they are all based on the 3D bead beating technology. So from the smallest one, which is the Mininis, where you can treat few tubes at the time, which is more dedicated for daily use, what we can say daily use, so few samples a day, up to through high throughput with the Precellys 24, when you can treat 24 samples of 2 milliliter tubes, and the biggest one, which is the Precellys Evolution, where you have very high versatility and a very high efficiency, and well, quite high throughput as well. So you can treat different size of tubes and different speeds, and you also have the cooling system, which we'll discuss later on. Associated to these instruments, we have a very wide range of consumables by mixing different kind of beads and different size of beads, different material, different size of tubes. So you can find a match with every kind of samples that you need to treat in your experiments. So what are the improvements that you can bring to your sample preparation thanks to 3D bead beating technology to the Precellys range? So as we saw in introduction, breaking the sample is not the only point. You need to break your sample, but also to get enough molecules of interest and also not damaging them. So this is what we are going to see. Getting enough material, of course, when you are working with very large amounts of sample, it's not an issue. But if you are working with very few material, very precious material, you want to make sure that you will be able to use this sample to get what you want and to answer the questions you need to answer. So in that case, a lab was making rice genotyping, which basically is not quite rare sample, but however, they were used to use mortar and pestle, and they got enough material from some amounts of rice. By using the Precellys, they used much less samples, so only a few rice grains, and they got the same amount of molecules, so allowing them to perform their genotyping, their rice genotyping. So they were quite happy with the fact that it was much faster because they were able to treat a lot of samples at the same time. Much more efficient than the mortar and pestle, as they were able to get the same amount of molecules in a few with a few grain rice, and it was much more easier, of course, because there was no pain to get the molecules. So this is the first aspect, and well, obviously the instrument is quite efficient for that. Another aspect is the quality of the molecules of interest. So getting your DNA or your RNA is not the only point. As I said, you also need to make sure that it's going to be usable for your final analysis. So in that case, a lab was making some studies on frozen tumors in order to make NGS of these tumors, and so they were working with well, basically a manual method as well, and they performed the same experiments with a Precellys. The goal was to extract the DNA, and as you know, for NGS, you need to perform a library. So the library of your sample needs to be done from good length fragments. So there is a real need of getting enough material with a good fragment length, and in that case, the minimal length was over 23 kilobase pairs. So the extraction was performed thanks to the Precellys, and they made a quantitation on on another drop and the tape station. Tape station graph is here at the bottom, and as you can see, we have a quite narrow peak representing the fragment length of the DNA extracted from the samples, and you can see that the medium, the average was around 34 kilobase pairs, which was over the minimal requirements. So in that case, it shows that this sample preparation with a dedicated lysing matrix and a dedicated protocol can be very efficient to perform NGS, for example. But it's true also for other techniques. Aside the technique itself, you also need to make sure that that the molecules that you are going to extract from your sample are not going to be degraded, which is the second challenge that we have seen in this part. And in the case of nucleic acids, for example, you want to make sure that your sample are not going to be degraded by outside components like DNAs or RNAs. So all the consumables that we provide are RNAs and DNAs free. Another thing to be considered if you are working with proteins is the action of proteases. So proteases are basically isolated from the rest of the cell, and the rest of the tissue usually are separated in normal conditions from the rest of the cells, from the other components. But when you make the homogenization step, of course, you break all the cell walls, and so you release all the components of the cells all together. And so in that case, the proteases can degrade the proteins that they will meet. So if your protein of interest needs to be integer during the final analysis, you are going to lose some signal, even if you get a very high amount of proteins of amino acids after the homogenization. So to prevent that, to prevent this denaturation or degradation more than denaturation, you need to you can use some chemical compounds called proteases inhibitors. So they will prevent the action of the proteases on the proteins, and so preserve the integrity of the proteins. So this is a product that we've developed last year, and that is available in our catalog as a lysing kit that we call protein safe lysing kit. So in addition to the beads contained in the tube, you also have proteases inhibitors that prevent the degradation of the proteins and ensure a good final analysis, like proteins activity or whatever, if you are working on that. Another challenge that you may face with 3D bead beating, which is the counterpart of the efficiency, is the heat elevation in the tube. As I said, you can homogenize almost any kind of biological sample, even the hardest ones. But this means that for that, you need to have a very high energy delivered. This very high energy will be translated by heat elevation, by the friction of the beads on the sample. And as you know, heat and biological molecules are not always friends, specifically if we are talking about RNA or proteins or very fragile chemical compounds. So you can get degraded RNA or degraded proteins. Same thing for your metabolites. What we made is that we developed a system which is called the Precellys that generates cold during the homogenization process in order to inject cool air in the homogenization chamber, so where the tube are shaken in the instrument, in order to prevent the heat elevation. It's based on a very simple process, which is the dry ice sublimation. And so dry ice, when it sublimates, generates and releases cold. So we use this phenomenon in order to cool the ambient air which is injected inside the homogenization chamber of the instrument. So by this method, we are able to control actively the temperature inside the homogenization chamber, and of course to prevent the degradation of the samples in the tubes during the process. So just to illustrate this aspect, it's an application note that we got from a lab which was working on microRNA expression profiling, and the goal was to make again a cDNA library for sequencing, and so they used the Precellys and the Precellys in order to perform this extraction. So here you have the protocol they used, so the kind of beads they were using, so 1.4 ceramic beads. You have the buffer they were using, so they also used a protecting buffer in complement of the cooling system, so in order to really protect the samples. And the sample used was hypothalamic tissue. So they performed the homogenization, and then they managed to perform the cDNA libraries of the microRNAs, which was a very good performance, as microRNAs are very very small molecules, a few base pairs, 16 to 20, 21. So you can imagine that they are very fragile, and it's quite difficult to get. So by using the method of homogenization of cooling plus the buffer, they were able to get this cDNA library and to sequence it. So here are just a few examples. If you want to find more, you can go and visit our website, where you can find more protocols, more application notes, eventually some product news. But you can also get in touch with us, so you can contact experts, and we will be very happy to answer your questions if you don't find any the interest information you are looking for on the website. So just to conclude, just to sum up, so with the 3D bead beating and the Precellys range, you can work in your lab, even in high safety laboratories like BSL-3. The bead beating is very efficient for a lot of applications, able to treat any kind of samples. We have some ways to increase the efficiency of the extraction, but also to ensure a good protection of the targeted molecules. And well, as I said it, the 3D bead beating has become a gold standard. We have a lot of users across the world who sometimes leave some comments on Select Science. So if you want to give a look to these comments, you can go, and you will be able to to see other experiences that scientists share with their Precellys. So just to conclude, just keep in mind that nothing beats Precellys. You will be stronger in your experiments with your Precellys. And well, if you want to get in touch with us, so you can go on the website, but you can also reach me by email by using this email address, or application specialist, Florian, who is based in the US. So here are email addresses. We have time now for question and answer sessions. So I'm just going to check if we have questions. I think I saw some. So the question is, if you want to preserve RNA, DNA, and protein, do you recommend using the protease inhibitor tubes? So there are different ways to approach this question. If you want to make just one homogenization and then split your sample in order to make DNA or RNA extraction on one side or protein extraction on the other side, I would recommend not to use directly the protease inhibitors in the tube, because, well, it's not interfering more than that with the DNA purification, but it's sometimes better to prevent that. So if you really want to make sure that you will have preserved proteins, I would recommend to split your sample in two different tubes, one for protein extraction and the other one for DNA and RNA, and then treat your sample for protein extraction with the inhibitors directly. So it's better to make two tubes, and one for proteins and the other one for DNA and RNA. But you can still make everything in the same tube, but in that case, I wouldn't use the proteases inhibitor in the same tube as for DNA or RNA extraction. I don't see any other questions. So maybe I can leave you just 1 or 2 minutes in order to write more if you want. Well, it seems that we don't have no more questions. So I'm going to thank you all for your attention and your participation to this webinar. Well, if you have questions after the webinar, do not hesitate to get in touch with us. Of course, we'll be very happy to answer you and to get in touch with you. So if you want some personal advices or things like that. And well, I wish you a pleasant day, and well, hope to talk to you soon. Thank you. Bye-bye.